Okay, guys, so continuing the food production topic and just quickly reviewing bread production and yogurt production, then we'll move on to fermenters. So our first step, we make the dough, as you can see by the picture here. We combine yeast with flour spelled wrong. We add water and some sugar for the process to happen. Dough is then kneaded, as you can see here. And we mix all the ingredients together and we leave it somewhere warm. The enzymes in the yeast break down the flour, again spelled wrong, to release sugars. These are first used in aerobic respiration. Aerobic meaning with oxygen. But as soon as the yeast runs out of oxygen, what happens? The yeast switches to anaerobic respiration and produces lots of CO2. That's what we want. And ethanol, which we don't really want. So the dough is left for a few hours for this process to happen. The dough then rises. Why? Because the CO2 is produced and the bubbles get trapped in. That's why we see some holes in our bread. It can actually get 4 to 5 times larger than its initial volume. The dough is then baked in a hot oven. The yeast are killed. That's why it doesn't continue expanding. And ethanol is evaporated. That's why we do not get drunk from bread. Now moving on to yogurt using lactobacillus.
Assalamu alaikum.
Who's that?
Fahad Salim.
Fahad. Hello, Fahad. Assalamu alaikum. You don't need your mic. You can just follow. I was actually just recording a video for you guys, but anyway, just follow. Did you watch the video yesterday? Was it?
Or crop
It was
farming?
about bread. It was about bread and yogurt.
No, I don't think.
You didn't?
Let me check.
Okay. Well, I'm just going over it now. We did do it in class, but we'll just go over it. We'll just go over the steps quickly now, yeah? So yogurt, how do we make yogurt? The first thing we do is sterilize. We know what sterilize means. Killing off any unwanted bacteria with bleach or steam. Then pasteurization, which means heating the milk up to 72 degrees Celsius for 15 seconds, which again kills any bacteria within the raw milk. Then we incubate the bacteria that we want, which is lactobacillus. We incubate it at 37 to 44 degrees Celsius. That's the optimum temperature for up to 6 hours. Then fermentation takes place. This is where the bacteria, this is the important part, the bacteria converts lactose sugar into lactic acid. And because the pH drops, this makes the milk solidify. That's why yogurt is different to milk in its consistency. Now, cooling it down, the yogurt is cooled down to about 5 degrees Celsius. Then we can add other things like flavors and fruits. So, Fahad, just briefly, this is the summary of what's happening. So we sterilize, we pasteurize, then we add lactobacillus, we incubate, letting it grow. This then converts, during fermentation, converts the lactose into lactic acid, and then we add fruit. So now this is our topic for today, fermenters. So microorganisms, as we've seen, they're very useful. We also use them in production of penicillin and insulin. Now, we need to grow them in something called a fermenter, this machinery here. We want to grow our useful bacteria, and we do that using this piece of equipment to hold a liquid culture of microorganisms, so it's in liquid form. The conditions inside we can control to make it optimum for the growth of bacteria. Fahad, what does optimum mean?
Maximum.
Maximum, is there a better word? Better words? Yeah, so the best, yeah? The best levels for growth. So in terms of temperature, nutrients, etc., we'll see in a minute. So you need to know this diagram for your exam. So number one, the fermenter must be sterile. What does sterile mean, Fahad?
Cleaned from bacteria.
Yes, exactly. So we do this by pumping steam through the equipment, through here at very high pressure, through this pipe here. Steam goes through. Then this stops contamination and the apparatus is now said to be aseptic, which means completely clean, sterile. Okay, the next step, number 2. The culture medium containing microorganisms and nutrients can be added. Here, this green sort of liquid, we call it a medium culture. Then this thing here, this black thing here, is called a motor, and it turns, and these are called paddles. Imagine like a fan. These things here kind of looks like a fan, and it stirs the solution, making sure everything's moving around. Okay, so then we put water in and water out. Fahad, why do you think we need water around the medium, not inside, around it? Why do you think?
To cool to keep temperature.
Exactly, yes, to control the temperature. Good. So water is used to keep the tank cool and keep it at optimum temperature for growth. Air is then pumped in if the organisms need to respire aerobically. So in order for organisms to respire, we pump in some air. As you can see, the air bubbles there. Then the next step, a special probes. These things here. Let me just go back. These things here. These things are probes, so it can measure the temperature and pH levels to make sure the conditions are again optimum and we can control them. So these probes are linked to a computer. Okay, so how can we investigate fermentation? Well, because we know yeast is a living organism and we know it can respire anaerobically at different rates depending on conditions. So we want to see the rate of respiration. So how could we investigate this? Well, here's the setup. What we do is we add this layer of paraffin oil. Why? To make sure yeast is not getting any oxygen. So this is the yeast. So it's blocking any oxygen getting to the yeast. We add sugar to the yeast for energy. And then hydrocarbonate indicator or lime water can be used to detect the amount of carbon dioxide being produced. We should know this from our respiration topic, hydrocarbonate indicator, and how it shows, depending on the color change, the levels of CO2. So now we can change different variables to see, or different factors to see the rate. Fahad, what does rate mean?
Rate mean per second.
Yeah, so how fast fermentation happens. And yeah, go on.
خلاص.
Yeah, okay. So we can see the rate of fermentation. How? By measuring the rate of carbon dioxide production. Basically how long it takes for the volume of carbon dioxide, for a given volume of carbon dioxide to change the color of the indicator. So we can count the number of bubbles per minute, basically. Similar to a photosynthesis experiment, if you recall. So we must remember to only change one variable in each experiment, so we can investigate the effect at changing that variable. So the variables we could change, for example, is temperature. The second investigation, amount of yeast. Third investigation, the type of yeast. And the next one could be pH, see the conditions of acidic conditions or alkali conditions, see how the yeasts ferment under those conditions, and also the concentration of glucose to see how the rate changes. We could also do the experiment more accurately using a gas syringe to measure the volume of CO2 produced. Okay, so that is fermentation there. So all we need to do is to measure the rate, all we need to do is either count the number of bubbles per minute produced and see how long the color changes of the hydrocarbonate indicator. That's it for now. Our next topic is fish farming. 1 second, it's just frozen. Just for the hydrocarbonate indicator, I just want to remind you. Fahad, what can you see? Can you see anything on your screen? Fahad,
No, I
still there?
can see. Yes, I
You
can
can't
see.
see anything. Okay. Okay, just give me a second. I just want to show you hydrocarbonate indicator. No. Oh. Is it here? Yeah. So just give me a second. Fahad, do you remember the respiration topic?
Yes.
So do you remember hydrocarbonate indicator?
Yes, it's the yellow one and red one.
Yes, exactly. So here it is, actually. No, that's glucose. Give me a second. I'm just trying to find the slide. Yeah, you remember? Okay. Let me just share this screen with you. Here, so if you remember this experiment, the respiration experiment, where we have living organisms, you can see how much CO2 is produced, or to make sure they're living, and the indicator they use is a hydrocarbonate indicator, and it will change color from red to yellow when carbon dioxide level increases because of respiration. Okay, so that's all I wanted to show you. I'm just going to end the video here and start a new one about fish farming. Okay, so join the next meeting. Actually, no, leave it. You can stay here. I'm just going to...
Okay, I'll wait.